Welcome to our review on monoclonal antibodies. So the first thing we're going to look at is what is a monoclonal antibody? And quite simply, these are monoclonal antibodies because they're produced by a single clone of cells. Now what that actually means is we're going to produce these in the lab by using a type of cell called a hybridoma. A hybridoma is a fusion of two types of cell, myelomas, which are cancer cells, and lymphocytes, which are white blood cells that produce antibodies. So once we've fused those two types of cells together, we've got a hybridoma, and they then produce these antibodies for us. What we do is we actually design these monoclonal antibodies to target a specific type of cell. And the way we do that is that they're designed to bind to antigens of our target cell and either kill it or prevent it from operating effectively, depending on the end result we're aiming for. So if we think back to what we looked at in terms of our white blood cells, we know that lymphocytes produce an antibody which is specific to a particular antigen. So we're just using that feature of our lymphocytes to then give us something that's able to target a specific type of cell. The next thing we really need to consider is how we produce them. So we're going to start off with some good old genetically modified mice. So we inject those GM mice with the required antigen, and the body of the mouse will then produce antibodies using its lymphocytes to that specific antigen. We then collect the lymphocytes, and then we fuse those lymphocytes with our myeloma or cancer cells to make the hybridoma. So what we have to do is carry out that fusion stage because the lymphocytes can't actually survive outside the body on their own. So if we didn't fuse them to the myelomas, they wouldn't last. And the reason that we use myeloma cells is that they can reproduce indefinitely. So that means that we can keep producing more and more of them for an indefinite period of time. As our hybridoma cells then reproduce, they form clones. And remember, a clone is an identical copy. So that means that each clone will produce the required antibody. And we can then harvest all of those antibodies that have been produced by these hybridoma cells. And those are the monoclonal antibodies that we can then use in a range of different things. If we consider what we can use these monoclonal antibodies for, the first thing is home pregnancy tests. So from about 2 weeks after conception, then the human body will produce this hormone called HCG, or human chorionic gonadotrophin. Now, what we can do is we can get monoclonal antibodies that bind to the HCG, and then when they do so, we can produce a colour change reaction. So that means that if we've got one of those pregnancy test sticks, we've impregnated a band of those monoclonal antibodies on there, so that when the urine containing HCG comes into contact with them, we get a line appearing on the stick that tells people that they're pregnant. A second use for our monoclonal antibodies is in detecting diseases. So we can actually use them as markers because they will be able to bind to a specific antigen and then confirm its presence. So a good example of this is in the diagnosis of prostate cancer. And a big advantage here is that it's going to allow for early diagnosis of the disease before we're necessarily getting too many symptoms, because it's going to bind to those antigens and show that it's present even when it's in very low quantities. The final use for our monoclonal antibodies is in treating cancers. So as we've already said, these monoclonal antibodies target specific cells. And when they do so, they can result either in the death of the cell or in the prevention of its proper operation. So if we were to actually attach a drug or a radioactive substance to the monoclonal antibody, then we could have a delivery system that will only go to those specific cells with the antigen on the surface. So we can target just the cancer cells with the radioactive substance or the drug. This is really important in cancer treatments because that means it's going to minimise the damage to the surrounding healthy tissue, which is obviously a big problem of most of our cancer treatments. Because cancer cells are just normal body cells gone a little bit crazy in their division, this means that any drug that's going to kill a cancer cell would kill normal body cells as well. So by having a delivery system that's going to be targeting just those cancer cells with the specific antigens on the surface, we can minimise the damage to the surrounding tissue and therefore reduce the side effects of the treatment. At the moment, we've got monoclonal antibody treatments that have been developed for 3 types of cancer, which are breast cancer, stomach cancer and bowel cancer. And there's still a lot more research that's ongoing for the other types of cancer to try to develop this as a technique for the other cancer treatments. Hopefully at the end of this video you can now describe what a monoclonal antibody is, you can describe how they're produced, and also described how they're used in 3 different areas.